Welcome to Study Loop.
For this question, we are taking a look at the lac operon genes. So this is a sequence inside the DNA that is responsible for producing the lactase enzyme, responsible for digesting lactose inside the bacterial cell. So this is going to consist of multiple parts. You have the operator, and you have the lac genes, such as lac-Z, lac-Y, and lac-A. And of course, at the beginning of this, you'll have the promoter where the polymerase is going to initially attach. So here in a normal process, the operator is responsible for turning the sequence on or off, meaning if the operator is bound and repressed, these lac genes will not be transcribed. Whereas if the operator is unbound and free, it will allow lactase to be made through the transcription and translation of these different genes. So here we are being asked to consider a certain mutation in one of these genes, such as, say, an addition mutation, or through a deletion or some other sort of mutation. These mutations do not have an effect on how the gene and DNA sequence is read. So regardless of how these genes look or their nucleotide sequences, as long as this lac operator is on, the polymerase is going to transcribe these three genes. What the mutation does, though, is the mRNA that is produced through this process is going to contain that complementary mutation as well. So here, the mutation inside one of these genes is going to be reciprocated to the mRNA strand itself. And of course, when the mRNA strand is translated, the proteins being made from the mRNA strand are going to also contain that mutation, as long as a different amino acid is coded. So here, the results of a mutated gene inside of this lac operon is going to result in a mutation in the protein strand itself. However, transcription and translation are still going to occur at the same pace with no problems at all, because the problem is only in the coding and the sequencing itself rather than in the function of this process. So here the best answer choice is going to be D, that the only result of this mutation is going to be in the function of the protein itself, rather than in the actual translation and transcription process, which should occur normally.